start. Welcome everybody. This week is an interesting week because we're going to talk about genetics and biotechnology. It's a pity it's only 1 week because it's something that I really like, but at the same time it stresses me because it is a lot of information and we only have 3 lessons to do everything. So it's a little bit complex in the sense that to understand it in a lot of depth, then you should spend way more time on it, but we don't have more time. We just have this week. So that's why it's a bit annoying because I'm really fanatic about this. I think it's a very, very interesting subject. And unfortunately, we can only see like the main things of these genetics and biotechnology things. And just by seeing the main things, I hope you understand a little bit the importance of this, because this is really, really the future. You know, the biotechnology, it's really the answer of a lot of problems that we have nowadays, as you will see tomorrow and on Friday. So what are we going to do today is we are going to here.
Amelie's having problems with her internet. I'm not sure if she
Okay.
could enter or not.
Yeah, well, she can connect if she has a chance. Otherwise, she can watch the video. I didn't put her in absence yet. So what we're going to do today is we're going to see how the DNA molecule is built and some interesting facts about it. I'm going to start with a little video that I think it's very, very interesting for us because we're multicultural and in fact, the DNA tells us a lot about ourselves, as you will see in the video. And we will see some brief history of how they discovered the DNA molecule and how this DNA molecule is copied. So that's really what I would like to do for today. Before we start, I just want to remind you that tomorrow is Changemaker Day, okay? I would love to do it with you together and be there and do stuff. In fact, I'm going to connect myself to certain things if tomorrow when I don't teach. Of course, I cannot cancel my classes, but you can see here the agenda of Changemaker Day. I asked you last week to inscribe yourself because I think it's important that you as students are invited to certain things, okay? I leave it completely up to you if you want to participate or not. It's not obligated, but I just want to give you the opportunity to be part of this if you think it's interesting. It has very interesting talks about civismo, the technology in service of the community. It has a lot of talks that I would very much like to listen to. For instance, this one you could do, Consciéntete con un turismo consciente. Mexico, it's a very touristic country, but basically what's happening is that the tourism is like a virus, I think. I mean, they take Cancun, nobody wants to go to Cancun anymore, they take Playa del Carmen. Then Playa del Carmen is completely saturated, let's go to Tulum. Now Tulum is really getting unsustainable, dirty, cenotes are contaminated. Okay, it doesn't matter, we still have Bacalar, let's go to Bacalar. And this is happening everywhere in Mexico. You know, this is not sustainable. So I think this is one example of things that could be very interesting. But check it out. You got normally, if you got registered, you got an email with the agenda of things you can follow. I suppose you have the afternoon free. There is a lot of talleres and more talks about a lot of things. So if you can, please participate. It can only make you smarter and more conscient about certain things. So let's start with our DNA molecule, guys. The DNA molecule, it's something that you all know. I'm just showing a 3D model. This is a DNA molecule. You know how it looks like. You see it in my background. You have seen it all your life, okay? But this is one of the most complex, if not the most complex biomolecule on planet Earth. You know, it's super complex. What you see here is just a tiny, tiny fraction of the DNA molecule. And today we're going to try to understand how this molecule came to be, how they discovered it, how it works, and the complexity of all of this. So before watching the video, guys, I want to give you some numbers, okay? Some mind-boggling numbers. somebody knows how many cells you have, the average human being. So I'm tall, I probably have a little bit more cells than I don't know, I've never seen you live, but I suppose Cecia, you're smaller than I am, I guess. I don't know. I'm not, I don't want to offend you, but at least, I mean, the bigger the person, the more cells you have, of course. But I'm taller than most of my students always, because I have 1 meter 1 meter 87. I'm almost 1 meter 90. I'm a quite tall person, so I have more cells, you know, because I'm tall. If you're a smaller person, you have less cells. If you're a fat person, you have more cells than a skinny person. So it's approximately. So how many cells do we have to give you an idea? Somebody knows?
A million,
I
like
think-
millions.
I think trillion.
Like something trillion.
Yeah, I think something trillion.
Yeah, it's true.
Around
It's in the trillion.
there.
It's about 37 trillion, according to the most recent science. 37 trillion. Well, that is really a very, very high number. Think about it. That's, you have more cells in your body than there are people on planet Earth. You know, we are only 7 or 8 billion people on planet Earth, and there is 37 trillion. So 1 trillion is 1,000 billions. You know. So think about that. Well, each of those cells, they have a nucleus. You know that, you know. Last week we talked about the plasma membrane. Today we are going to talk about the nucleus. And in that nucleus, we have DNA. We have seen that already. And now we're going to investigate and talk about that DNA, only the DNA. So how long is the strand of DNA in each of those cells that you have? Somebody has an idea? So you have 37 trillion cells, and each of those cells, except the red blood cells, blood cells, they don't have a nucleus. They don't have a nucleus because they have a specialized function, which is carrying oxygen. So it was necessary for evolution to get rid of the nucleus and put hemoglobin in the red blood cells. Otherwise, it would be too packed. So there's one of the only exceptions. Red blood cells, they don't have a nucleus, but all the rest have. So red blood cells, they don't have DNA. Only white blood cells have DNA. Anyway, how long is the DNA in the nucleus of our cells? In our skin cells, liver cells, muscle cells, nerve cells, pancreas cells, every cell, eye cell, brain cell. Just give a guess. No idea?
A meter.
Yeah, it's 2 meters long, in fact, 2 meters long. And now, if you would take the DNA out of each nucleus, the 2-meter DNA out of each nucleus of your 37 trillion cells, and you would connect them all to each other, the DNA would have the length of twice the diameter of our solar system. Our solar system is really, really big. We're talking about 287 billion kilometers. I mean, it's really, really long. And to put that in perspective, it's 1.5 light years. So it's very long, guys. It's really, these are numbers that it's very difficult to conceive, but that's what we're talking about. That's how complex life is. Okay, guys, I want to share with you now a video. Don't cry. It's a very nice video. And I think it's important that you watch this video because we are multicultural. There is a lot of racism. There is a lot of problems in the world, ethnic issues of people. And this video gives you a complete different perspective on DNA. So this video is about DNA and what it means. So just watch it and then you see why it's important for us. Let me share it with you. There we go.
It's easy to put people in boxes. There's us.
Sorry. Sorry guys, wrong video. It's also a nice video, but it's not the one we wanted to see. It's this one. Sorry, I put the right one now. It's the one that comes next. In fact, you should see it too, but we don't have so much time. The other one is also really nice.
I'm proud to be English. My family have served and we've defended this country and we've been to war for this country.
I'm really patriotic about Bangladesh.
Well, I am 100% Icelandic, yeah, definitely.
This is a Kurdish wedding with my mom in the traditional Kurdish clothes.
We're just proud black, so that's it.
Yeah, I think we are probably the best country in the world, if I'm honest.
Think about other countries and other nationalities in the world. Are there any that you don't feel you get on with well or you won't like particularly?
Germany. Yeah, I'm not a fan of the Germans.
You might think they're a little bit...
Particularly India and Pakistan, probably, because of the whole, the conflict.
Because I have this side of me that's like, that hates Turkish people. Not people, but the government.
But French?
No.
We're just the best, it's just fact.
I'm more important than you. I don't know you, but in my opinion, I am strong and I am more important than a lot of people.
How would you feel about taking a journey based on your DNA?
Yeah, I feel very intrigued.
What could you possibly tell me that I don't know?
So do you know how DNA works? So you get half from mum and half from dad, so 50% from each of them, and they get 50% from their parents, and back and back and back. And all those little bits of your ancestor, they filter down to make you, you. I need you to spit in this tube for me. You spit up to the little black line.
That's a lot of spit.
Right, the story of you is in that tube. What's it going to tell me?
It's going to be, oh yeah, you're French, and wait, your grandparents are French, and wait.
100% Bengali.
Solid Iraqi.
I'm Cuban.
You're going to tell me that I'm English? I've told you.
Jay, can you come down and join us?
I'm a little bit nervous, I have to say.
So you ready to find out your results? Will you read it out to us, please?
Wow, look at me. Oh my God. Oh, wow. Shit, I didn't expect that.
All of them.
Whoa! No.
Caucasus, which was Turkish?
Yeah.
Eastern Europe, Spain, Portugal, Italy, Greece.
I'm 32% British.
What? Great Britain, 30%. Can we 5% German?
I'm Irish.
Yeah.
So I'm a Muslim Jew.
Great Britain, 11%. Are you sure these results are mine?
Eastern Europe?
Iceland has definitely moved closer to Europe now.
I'm going to go a bit far right now, but this should be compulsory. There would be no such thing as, extremism in the world if people knew their heritage like that. Who would be stupid enough to think of such thing as, a pure race?
In a way, we're all kind of cousins, in a broad sense. In a much more direct sense, you have a cousin in this room. Turn around and guess who it is. Waj?
Yeah. What's that?
Why don't you come down here and meet your cousin?
God, this is our best.
Did you know that?
I did no idea. This is like my heart's pounding right now. I swear to God. We met.
Jay from everywhere, but every to this.
I'm a real man of the world.
Wow, that's beautiful. Thank you.
So would you like to travel to all of these places?
Yeah. Hell yeah.
Oh, yeah.
So what did you think of this?
Well, that some people don't realize the amount of heritage that they have inside their body.
Yeah, I think it's a very beautiful video, and in fact, I'm always curious about this. You know, if I would do this on myself, I think I would see, okay, I'm a bit Dutch, I'm a bit from Sweden, my mother is French. I think I would be very much in Europe, maybe a little bit boring, but you guys, you would be super interesting because you think, I mean, I don't want to create any offense, but all of you think that you're Mexicans, and of course, on paper, you are Mexicans. But if we would do this with you guys, it would be very, very interesting because many of you, look how white you are. I mean, you could be even my family, you know. If we would do this DNA test with you, Ana Pau, Isabella, Itzel, Gustavo, even Javier, Gael, definitely. All of you, if you would do this, you would see that you're possibly more European than you are Mexican, because if you would be 100% Mexican, you would look like indigenous people from Mexico, and most of you are definitely not. So especially for you, I would love to do this. I mean, I don't have the means to do this, and because it's quite expensive, it's about $200 each. But it would be a very interesting experiment, especially on you guys, Mexicans, to see where did you all come from, because I mean, your descendants from Spanish colonizers or even American Irish people that came to the US. I mean, I really don't know. It would be interesting to see, but what we would see is that you're not very Mexican, I would think. I think you would be more European than you think. So that's really nice. And also, I like the moment that the French woman says like it should be obligated, because really it would get rid of a lot of racism, because it's so ridiculous to be racist about certain things and thinking you're better or worse than somebody else when we are in fact all the same, you know. I mean, we're all made of the same material, our DNA is the same, it's just certain physical colors and physical structures might be a little bit different. So guys, let's have a look to this DNA and how they discovered it, okay? Let's go through the history a little bit, where this genetic material comes from. First and all, I want you to be relaxed, okay? There is a lot of information here. Might seem very complex, but I'm gonna tell you really what you have to know. You don't have to know all the details. It's really not the idea. For me, the objective is that you understand what the DNA molecule is, where does it come from, and how can you duplicate it, okay? So let me start telling you how this happened. Well, the first person, we have to go back to 1869, was Friedrich Miescher, okay, Miescher. And Miescher, he was a good scientist, but he had no clue about genetics, okay? In those days, they had no idea why you would look like your mother or your father. They had no clue which kind of molecule would give those characteristics, okay? Because of course, they already know since thousands of years that sons and daughters, they tend to look like their father. So there is something, you know, there must be something which causes the children to look like their parents. He didn't know anything about this. He didn't know which molecule was it, but he found a molecule in the nucleus of cells. Okay, he didn't identify that as being genetic material, but he was basically the first person that noticed that there was an acidic substance in the nucleus, and he was the one that first discovered this molecule. Okay, the existence of the molecule without knowing what it was really doing. Okay, that's Michert. Then we go to Griffith. Okay, that's already 40 years later. What Griffith did was like an experiment. Okay, look at these mice. What he did was, you have bacteria, okay, blue ones and red ones. Okay, non-virulent means that they're harmless. Virulent means that they're harmless, that they cause, they damage. Okay, so what happened is that what Griffith did, he injected a mice with bacteria, and the mice would be fine. Okay. Then he would inject a mouse with other strain of bacteria that are dangerous, like pathogenic bacteria. And these bacteria, they kill the mice. Okay, everybody with me so far? Very simple. Okay. So what he did next was, okay, let me cook the bad bacteria and let me inject these bacteria in the mice. So what he did, he cooked the bacteria and he injected the dead bacteria in the mouse. And what he saw was that the mouse stayed alive, like you can see here in the third picture. But then what he found out, he combined alive, harmless bacteria with dead, dangerous bacteria, and he would inject both in a mouse, and what happens is the mouse dies. Okay. So Griffith doesn't understand what happened. He just did the experiment and he wrote down what he saw. So what basically happened is that the good bacteria, they took the DNA of the dead bacteria, and it started to be, it became a part of the good bacteria. So the good bacteria transformed into pathological or virulent bacteria that killed the mice. Okay. Why is this experiment important? Because this indicates that there is something in the cell that can transfer characteristics to other cells, okay? And that's called the transformation principle. That's what Griffith discovered. He didn't know what he was looking for. He didn't know which molecule it was, but he did discover that there is something in the cell that has information that can be passed to another cell, and that then the other cell takes the characteristic of the other one, you know? Everybody gets that? Okay, very well. So that's basically what Griffith did. And then we go to Avery, MacLeod, and McCarthy. So basically, they continued a little bit with the work that Griffith found out, okay? Griffith found out the transformation principle, but Griffith had no idea which molecule was causing these characteristics, which is the molecule that causes the mice to die. So what did these three scientists do was they would add enzymes that destroy a certain molecule in the mix to identify which molecule it is, okay? So certain enzymes. So these enzymes are called DNAs and RNAs, okay? And basically what they found out is that not protein and not RNA are responsible for the characteristics. So they only stayed with one remaining molecule, and that was confirmed by Hershey and Chase with another experiment that DNA was the molecule that has the characteristics of life encoded in it, okay? So what Hershey and Chase did in the 50s, they used bacteriophage. I don't know if you have heard of bacteriophage, but it's nowadays something really important. It's a virus that can infect a bacteria with its DNA. So nowadays, those bacteriophage, we use them to alter bacteria. I give you an example. What we do, for instance, is we are developing with biotechnology bacteria that can eat plastics. to biodegrade plastics. But these bacteria, they don't exist in the natural world. So what they do is they take certain enzymes or certain pieces of DNA, they put it in the bacteriophage. The bacteriophage injects it in the bacteria, and the bacteria starts to do a job that they normally would not do. Another example of what we do already today is human insulin. You all know diabetes people nowadays, right? You all have somebody in your family who is diabetic. Well, like many years ago, those people, they would inject pig insulin in order to stay alive. But pig insulin is not the same as human insulin, and created certain reactions. Nowadays, you can be diabetic person and live perfectly fine. Don't forget that even 50, 60 years ago, people would die of this illness. It's a very serious illness. Nowadays, you live with it and it's just like a flu, or yeah, I'm diabetes, nothing bad is happening, but it's not the case. But the reason why these people nowadays can live so good is because they have human insulin. And that human insulin is not made by humans, it's made by bacteria. What we did is we take bacteria, we put a human gene in it that produces insulin, and we use bacteria to produce that insulin with the principle you see here, okay? So what is this experiment explaining us basically is they would infect bacteria with bacteriophage, and they would see that the bacteria would have different characteristics, confirming the fact that DNA indeed is the molecule that has the genetic code inside it, you know, that it's DNA coding for life, okay? So how does DNA work, guys? You all know it's a molecule, right? It's about 2 meters long, and it's made of nucleotides or base pairs, okay? And those bases or nucleotides have 4 names, okay? There is 4 codes, okay? Think about a computer. How does a computer work? Somebody knows?
With codes?
Yes, but what kind of code is it that a computer has?
Binary?
Yeah, it's a binary code. So basically, we're on Zoom right now and you can see my face, but this is all artificial. It's all made by ones and zeros, a binary code, okay? So that's how our technology works, with zeros and ones, 1, 1, 0, 1, 0, like this. Well, the genetic code, our code that we have in our DNA, which we call the genetic code, is not made by ones and zeros. It's made by 4 things instead of 2. And those 4 things are A, G, T, and C, okay? And these are nucleotides, and we call them adenine, guanine, thymine, and cytosine, okay? And this was already discovered in the 50s. This is the building blocks of DNA. Well, there is something special about those codes, about the A, G, T, and C. Well, they discovered, so the A and the G, you can see here, they have like a double ring on top of it. You see here, the orange and the green. And then the T and the C, they have a single ring, the blue and the red. So the one with the double rings, adenine and guanine, we call those ones purines. Okay, it's just the name. And the ones that have a single ring, the T and the C, we call them pyrimidine. Okay, pyrimidines. Okay, this one and this one. Okay.
Repeat those last names, the things and the one.
Yes, Amelia, you have to know the name of adenine, guanine, thymine, and cytosine, and you have to know that adenine and guanine are they're purines, and the pyrimidines is thymine and cytosine. Okay. Well,
is it related? Is this related with the form of the nucleic acid?
Yes, dear, definitely. I go there in a second. It's these things that make the shape of the molecule itself. Okay, there is something special about these molecules, okay? They discovered, I'm not going too much into detail, like Erwin Kargav, they made discovery about the composition of DNA, okay? They discovered that the proportion of A and T and the proportion of C and G was roughly the same. it was 40% of thymine and adenine, and it was 60% of cytosine and guanine. So it means that means that there is more or less the same amount of cytosine and guanine as there is as thymine and adenine. So they figured that if this is the proportion, then it means that A combines always with T and G always combines with C. And this is really groundbreaking, because this helped Watson and Crick later on to come with the molecule itself. So here you can see the numbers. You can see here the A and the G and the C and the T, okay? And then here you have the ratios, okay? The ratios were more or less the same. They still didn't have an idea of how the molecule looked like. They just identified those nucleotides, but they really didn't have a clue until Franklin came along. And what she did, she sent the X-ray through DNA, and she would get a diffraction pattern. And this is the picture they took. And this is, in fact, the first picture of DNA ever taken. And it's this picture that was really, really important, because it gave us an idea of how DNA would look like. And finally, in 1953, it was Watson and Crick that discovered that the DNA looked like this, like how I started the class, and that it looked like the double helix, okay? And they were also the ones that discovered that the G would always combine with the C here, and between those two things, you would have hydrogen bonds, and the T would combine with the A. And I go quickly back to that. And why they combine has to do with the nature of the molecule, okay? The chemical composition of the molecule creates repulsing and attracting forces. Some of those forces that attract, we call them hydrogen bonds. And this makes it that A can only bind to T and G can only bind to C. And this is very, very important, because when you have a DNA strand, like you can see it here theoretically, you see T is combining with A, G is combining with C, okay, and so on. And basically the code is like this, you know, T, G, G, T, A, C, A, G, G, okay? And it goes on for millions and millions and millions of base pairs like that. So how does the DNA duplicate? So that's in fact the first thing a cell does. When it decides to divide, it's gonna copy its DNA. When it copies its DNA, this is what we call DNA replication, okay? And I'm going to explain you a little bit how that works. So before a cell divides, the first thing a cell does is copying the DNA. Once the DNA is copied, then the cell splits into two. This process we call mitosis. It's a whole other chapter. We are not going to see that today. What we're seeing only today is how the DNA is copied. And why do we see it immediately with the structure? Because it helps you understand how the DNA molecule is built. Okay, like I said, C and G always combine. Now we have enzymes that open the DNA molecule, the double helix. So they unwind the strand. So you get one strand on each side. Like here you have one and there you have one. And then inside the cell can produce or can absorb or ingest these nucleotides. And when the enzyme opens the molecule, you see that A becomes free and T becomes free in here also. So what happens basically from the moment it opens, it fills up with the opposite base pair, with the opposite nucleotide. So this A will combine with what?
G.
A T. This C will combine with a G. This A will combine with a T. And you understand, like you can see here below, in fact, you get two identical copies. And this is the beauty of life, because this mechanism works so well that you can make perfect copies of each other just because this molecule only binds with that one. The A only binds with T, the C only binds with G. And what you get basically is you get two new strands of DNA. Okay, that's how the replication works. And it's of course way more complex than that. But what you have to know is just that we have certain proteins, which you can see here, the DNA helicase, okay? This is the enzyme or the protein that opens the DNA strand. And then we have a DNA polymerase, which fills up the DNA strand, okay? This looks a bit different than this one, but I'm not going to explain that because it would make it unnecessarily complex. Here, they instead of copying from here to here, they copy the other way around because otherwise you would get the inverted molecule. But don't forget about that. It's not important. It's just for us important to know that we have a DNA molecule that can be easily copied because it can unwind itself thanks to a simple enzyme. And then it can just be filled up again with nucleotides that are already present in the cell, and it can rebuild a complete new strand with almost 100% perfection rate. Okay, it's 99.99999 error mistake. So this is a very, very correct way of copying. Okay, so
I have a question.
Yes, go ahead.
The green thingy that we can see, that's kind of the DNA, is it the RNA?
No, RNA comes for next lesson. We're not talking about RNA yet. We're talking about DNA replication. So like I said, DNA has to be copied. Whenever a cell wants to duplicate, it needs to copy its DNA. What you're seeing here is DNA copying. Okay? It looks very similar than RNA, but RNA, we're going to see that tomorrow. That's a little bit different because RNA is a single strand. It looks like DNA, but it's not a double strand. It's a single strand. And
teacher, doesn't the RNA also have a U instead of the T?
Yeah, exactly. It has a uracil instead of a T, of a thymine. But we see that tomorrow. And the RNA, its function is to produce proteins. It has nothing to do with copying the cell. So this is basically everything I wanted to explain today. So here, this is for tomorrow. A little bit about the history, how the DNA molecule came to be, how scientists think about that. They were thinking always that proteins were the important molecules and that they would carry the characteristics of life. But it was not the case. It was a complete other molecule. I have a very nice documentary about it. If I recommend you to see it, I'm not going to oblige you, but I recommend you to see it. It's called the DNA helix. And it's a little bit about the history, and it could help you to understand a little bit more about this. If you're interested in it. Normally I make it obligated, but this year not, because we have other activities, because we're online. So if you want to see it, see it. I don't know if there's many documentary watchers amongst you, but it's very nice. In fact, I want to recommend also, have you seen the documentary A Life on Earth from David Attenborough? It's a new documentary about David Attenborough. He telling us what we did to the planet and what, if we don't act, what will happen. It's very striking. I think it's on Netflix. I also recommend that one. But to stay with the topic of the class, I share it with you in the WhatsApp group, the DNA helix model, if you would like to watch it. Any questions so far? If not, this is it for today, and we continue tomorrow with how proteins are made and how RNA is how RNA is formed from the DNA.
Can you show me the first slide, please?
Sorry?
Can you show me the first slide of the first person?
Can you share
Okay, first things.
this presentation with us, like in Canvas?
Of course. Yeah, of course. I share it with you. A ver. Which slide was it? This one?
First one.
Friedrich Miescher, this one. Okay, I'm gonna put this. I put this online for you. You can read it, and you just have to know the big lines. What did Miescher do? How did DNA come to be? How is it replicated? What are the building blocks of DNA? You really don't have to know the details. For me, it's just that you recognize it, and if you're interested in genetics, and you're going to study medicine or biotechnology or genetics in professional, you will see this in a lot more detail.
I just have a question before the class finishes. And for the core activity, how can we make sure it's our topic, or are they going to tell us something?
I sent to most people an email telling them it's okay if they selected their topic. So you should have chosen by now. And normally, if you selected the topic, it should be okay if nobody else has it. So
if you sent an email saying okay, it means that no one else has it?
yeah, exactly. I answered to you, Gretel. I think I remember that you wrote me, and you had something about coffee in Saudi Arabia. Was it you?
No, I think it was avocado in Morelia.
Okay.
Michoacan.
Yeah. If I said okay, it's okay. Go ahead, and you can do the investigation of what you would like with your group. Okay, so yeah, that's it for today, guys. Tomorrow we will have a small activity as well for a homework to do, and then on Friday we do a small quiz about this, like always. And don't forget that next week is already second partial. It goes fast, huh? We have 2 months left, and it's already Christmas. I mean, the class is finished, so we have 2 more months, and next week we have the partial. So see you tomorrow, guys, and take care. Okay.
Thank you, teacher.
Thank you. You're welcome.
Thank you,
Bye-bye.
teacher.
Thank you very much, teacher. Have a good day.
Bye.